Hi everyone, my name is Xiangfei Hong from Shanghai Mental Health Center. The title of my talk today is A Functional Subnetwork During Response Inhibition Reviewed by Network-Based Statistics. Previous studies have consistently shown the activation of a series of brain regions during response inhibition tasks, such as the stop signal task. Here is a figure showing the brain activation patterns from a meta-analysis which included 108 stop signal experiments. Based on such results, a large-scale functional neural network has been proposed as the neural basis of response inhibition. However, because such functional neural network hypothesis was primarily based on the contrast of neural activation between inhibition and non-inhibition trials, it still remains unclear that whether these brain areas are functionally connected during the inhibition task. The figure showing on the right is another study showing that response inhibition might involve the connectivity between the right inferior frontal cortex and other dorsomedial prefrontal and lateral frontal parietal cortex. However, the seed-based approach used in this study cannot reveal the whole brain connectivity patterns during response inhibition. So in this study, we tried to examine the whole brain functional connectivity pattern during response inhibition using a network-based statistics approach. To this end, we analyzed functional MRI data in a stop signal task from 104 healthy participants. For a validation purpose, we first ran a traditional GRM analysis to look at the neural activation during response inhibition. For the task-related functional connectivity analysis, we first estimated the single-try responses using the least squares signal model. After that, task-related functional connectivity was estimated using the beta series correlation method according to the AAL template. Network-based statistics was then used to identify clusters of functional connections. So basically, initial t-tests were applied to each of all possible links. We could get a p-value matrix representing the possibility of accepting the null hypothesis for each link. A set of super-threshold links with p-values less than 0.001 was then selected, in which connected links were identified as topological clusters. The significance of the identified clusters was tested using permutation test. Let's first look at the activation results from the GRM analysis. As shown in the figure and table, the contrast between successful stop trials and go trials revealed a series of brain activations. Overall, such activation patterns are consistent with previous research. Now let's look at the connectivity results. The NBS approach identified a cluster of functional connections that show significantly increased connectivity strength in the successful stop trials compared with the go trials. As shown in the figure, this cluster consisted of 33 links between pairs of 22 nodes. This table shows the details of the 33 links between pairs of 22 nodes in the functional subnetwork. We further ran a control analysis to rule out possible influences of the participant's age and head movements in the scanner. Head movement was quantified using the frame-wise displacement. The left panel shows the NBS results without the regression of the H and FD, which have been shown in the previous slides. The right panel shows the NBS results with the regression of H and FD. Overall, the two results are very similar. We also ran another control analysis to examine the effects of template selection by running the same NBS analysis based on power template. We can see that the two results are somewhat similar. So in conclusion, by using the network-based statistics approach, our results suggest that those brain areas that have been identified by previous research are functionally connected as a subnetwork during response inhibition. Therefore, the current studies pose a large-scale functional brain network basis for response inhibition. Thanks for your attention.